Hello, welcome to the next A-level lesson from Conscientious Biologist. This one's on enzymes. It's the first in just 2 lessons. This one looks at the mechanism of enzyme function, substrate specificity, and then the lock and key theory versus the induced fit theory. Now, most of this is pretty much GCSE level. There's a few terms, a couple of little bits of extra that's on there, but we'll go quite quickly on this because it should just be a review of really essential GCSE knowledge. As always, please do subscribe to my YouTube channel if you haven't done so already, and do check me out on Facebook and Instagram. Thank you. So let's start with enzyme formation, which is just a review really, because remember, enzymes are just proteins. Now, we've covered this in the previous videos and it's such an important bit of biology. So please do make sure you've gone back and you've watched DNA biochemistry, protein biochemistry, protein synthesis 1, 2 and 3, ideally mutations, which is basically how things can mess about with protein synthesis. But on this slide, let's really quickly recap some of those basic details. So we start with DNA. DNA we covered in DNA biochemistry. Here's a screenshot from that video. Go back and find it if there's anything that you don't think you fully understand about DNA structure. Because when DNA gets read by transcription, it creates pre-mRNA. Again, transcription, this should look familiar if you've watched that video. If you don't fully understand transcription, go back and have a look at it. As well as the transcription process in protein synthesis 3, we looked at a comparison of DNA and RNA, so you can see what the difference is between DNA and the pre-mRNA. Pre-mRNA then undergoes post-transcriptional modification to create mRNA, as you can see in this screenshot from protein synthesis 3. Have a look at this, have a quick read of it. If you're looking at that and thinking, I don't really know that, go back and watch the video. We're not going to review it now. mRNA can then undergo translation to create the polypeptide, which we covered in protein synthesis 1 and then reinforced in 2 for translating it for yourself. And then in protein biochemistry, we went over the amino acid bonding to form polypeptides in that protein biochemistry video lesson. So go check that one out if you haven't already. Then you get the final stage of protein folding to create your final finished structure. We covered that as well in protein biochemistry, and it's this either tertiary structure or quaternary structure that would give you the final finished shape, and shape determines function. Okay, so these are the really important stages in creation of an enzyme, and in the exam they're a bit sneaky. Sometimes they just say, explain how enzymes are formed. And people panic and they go, oh, I've never studied how it was formed. Well, of course you have. It's just the examiner's sneaky way of asking you to describe protein synthesis, which is just that over on the far side, often referred to as the central dogma, the whole process of going through it. But of course, we're not just talking about any old protein, we're talking about an enzyme. So let's switch that at bottom. And remember that enzymes are globular proteins. And we talked about the difference between globular proteins and fibrous proteins in a previous video. Okay, now think about when that's folded up. You might get a structure that looks like that on the far side. It wouldn't, but as a diagrammatic structure. You can see the polypeptide strand, the long blue one, all highly and intricately folded into that shape. Now, when we're trying to reproduce that diagram, we don't try and draw that every time. It's too complex. We just imagine we can draw around the outside and imagine it as a more solid 3-dimensional shape. So we end up drawing things like that. That's a fairly standard and hopefully very recognizable diagram of an enzyme. That's what we're going to be using through the rest of this video. So let's look at how an enzyme works. And again, this is pretty basic GCSE, but we might be adding some terms that you may not have covered. So you've got your enzyme. The key part of an enzyme you'll remember is the active site. That's basic GCSE stuff. That's taught in like year 7 as well, I think. So you've got the active site that acts like the mouth. You need to have something that's going to fit into that. That's the substrate. Now note, the active site and the substrate are complementary in structure. Just like DNA-based pairs are complementary, doesn't mean they're identical. It means they fit together. Okay, almost like opposites, like a lock and a key. The key must be the correct shape, but we'll talk about that in another slide. So the substrate there can move into the active site. If it does so, they'll form an enzyme substrate complex. The enzyme will break down the substrate by lowering the activation energy needed for the chemical reaction to metabolize it. That's a really important term. It lowers the activation energy. In chemistry, what we're talking about with that is that the activation energy is what needs to go into a reaction to break the bonds so that you can then reform things in another pattern. So you lower the amount of energy that needs to be put into it. That's what enzymes do because they are biological catalysts. Now, of course, I've got my diagram going across the screen here, but it can also go in the other direction. So if I put the same diagram below, but if I turn the arrows around, then we can call these closest to me the substrates, and they could build up to a single product right over there. So we've got two different mechanisms here. We've got one at the top there, where we're breaking one substrate into two products. But down the bottom here, we've got where you're joining things together. Now, there's names for this. Enzymes that break things down are called catabolic. Enzymes that build things up are called anabolic. Now, the combination of all the catabolism, that's the catabolic reactions, and anabolism, that's the anabolic reactions, when you combine all your cells, catabolic and anabolic reactions, that's what gives you your metabolism. So catabolism plus anabolism equals metabolism. Those should be familiar from GCSE, but they might not be. So take a screenshot of this if you're going to find it useful. Now, I spend quite a lot of time at GCSE explaining what substrate specific means, but I think it's pretty obvious really, so let's go through it quickly. If you look at that enzyme and that substrate that we had from the previous slides, you can see that they fit together. That enzyme is specific to that substrate, so we say it's substrate specific. Only that substrate can specifically fit that enzyme. If I've got other shaped substrates, for example these ones, look, then for the square shaped substrate, I obviously need an enzyme with an active site that's going to be complementary to that. So as it's written here, look, the shape of the active site and the shape of the substrate are complementary. They must be if it's going to be specific. That can fit into that one. But similarly, if I want a triangle that I'm going to break down as a substrate, it's got to fit into that enzyme. Each one of those enzymes is specific to only one substrate. Now that's really, really important for cell differentiation and for making sure cells carry out the roles that they're supposed to. Because if one enzyme can only do one job, then if a cell wants that job done, it just creates that enzyme and not other enzymes for jobs it doesn't want. So enzymes are substrate specific, which means a cell can be very specific in which enzymes it creates, which allows it to be very specific in which metabolic reactions it performs and what it does. It only makes the enzymes for the jobs it wants done within the cell. Now, if you think about how it does that, think back to the video that hopefully you've watched of mine on cell differentiation. So I'll pull up a screenshot of that now. I'll get rid of it in a minute so you can see this paragraph I've just brought up. But hopefully this looks familiar. And we talked in this video about how the DNA strands and the histone proteins that they're wrapped around can be methylated or acetylated or demethylated and deacetylated to flip the chromatin form between heterochromatin and euchromatin. If it's euchromatin, that means it's nice and open and can be read. So if a cell has enzymes it wants to be read, it would make sure the genes are kept as euchromatin, nice and open. Any genes for enzymes it doesn't need, it would bundle up into heterochromatin so they can't be read. So by controlling the state of its DNA, which form of chromatin it's in, it can control which genes are available, which enzymes can be transcribed and translated and hence expressed into proteins. And if they're expressed into proteins, then they can do the jobs for that specialist cell. So by controlling the enzymes and all proteins, of course, not just enzymes, that it produces, a cell can differentiate and become specialized. So substrate specificity, very tightly linked to cellular differentiation. Make sure you understand that. Last thing, and this is very quick, but it's going to link up to something in the following video. We're going to look at two enzyme theories, the lock and key theory versus the induced fit theory. Now, you should be familiar with the term lock and key theory. On the previous slide, I brought up a picture of a lock and key to try and make it really obvious that the substrate has to fit into the enzyme. So as we've talked about, there's my enzyme, there's my substrate. My substrate's going to move into that active site. It's going to fit perfectly, just like a lock and a key, okay? If they fit together really perfectly, that specific key could open that lock, that specific substrate will fit into that specific enzyme and do that job, do that little bit of metabolism. But in actuality, when you look at the data, when you study enzymes, this doesn't quite work out because this would have to be 100% complementary, no give or take, no tolerance of any mutations, no tolerance of any fluctuations in temperature that could affect the folding. Okay, it's got to be 100% right or it won't work. That wouldn't have been good from an evolutionary point of view. So what we get is this second theory that's induced fit, where if you look at this enzyme I've put up here just now, look at the active site. It's not quite right. If I move the substrate in there, you can see that there's a little sort of hollowed out gap. It doesn't precisely fit the substrate, but that's okay, because as long as it's almost a perfect fit, which you can see it is almost a perfect fit, then what will happen is it can be induced to fit, and the enzyme can kind of shift, and you'll see that where those arrows move, the protein can just kind of just warp a little bit and just go, yeah, all right, that'll fit. The equivalent would be is if you look at my hand, if I make that shape, that would be 100% fit there. That's like lock and key. If this was slightly narrower shape, look, it can almost slightly force its way in and induce the fit. Okay, so this is induced fit theory that shows that the active site only has to mostly match or be complementary to the substrate to be able to form an enzyme substrate complex. A fit can be induced. Okay, it can be made to fit in there. Now this makes more sense with the evidence of how enzymes behave, and we'll talk about that a bit more in the next video, but just be aware of these two theories, lock and key theory and induced fit theory, and why they differ from one another. So hopefully everything in this video was very familiar. It's very much an extension of GCSE. There might have been a few new key terms. Induced fit theory would have been new, but the key thing here is your A-level knowledge must be based on really solid A-level foundations. So now when you're explaining enzymes, you need to be explaining things about polypeptides, folding, amino acids, the tertiary structure, the ionic bonds between the R groups of amino acids, which can allow that folding to create the specific shape of an active site. You've got to get your A-level terms into your explanations when you're explaining it. A GCSE level answer is not going to be good enough to get you marks in your A-level exams. So make sure you've gone back, watch those previous videos, you've got all the detail and you can fully explain this. From here you can go to enzymes 2, which are factors affecting enzyme function. Again, not dissimilar to GCSE, but we now need to explain them with more detail, and there's an extra one as well that goes on top of this. Okay, head to that one next. As always, if you haven't done so already, please do give this video a like. If you find it useful, subscribe to my channel, and do have a look on Facebook and Instagram for additional supportive material. Thank you.